There are also other pathways triggered by the activation of the mu-opioid receptor, for example, phosphorylation. So the mu-opioid receptor contains serine and threonine, which contain accessible hydroxyl groups, so they can be phosphorylated by these G protein receptor kinases, so either 2 or 3. And there are many others, but this is just the one in the example. And this leads to recruitment of arrestins, which are proteins which bind to these phosphorylated regions. And that leads to internalization by endocytosis. So it forms, the membrane kind of pinches off and forms this endosome, this kind of vesicle. And then downstream of this, there are other pathways like the MAPK or mitogen activated protein kinase cascades, but we'll talk about those later. And then there's also protein kinase C phosphorylation, which is activated by the active G protein heteromer, and that phosphorylates the receptor in other places. And then finally they're recycled back into the membrane bilayer, so the endosome just fuses with the external membrane and it's recycled back onto the membrane without this phosphorylated region. But you will notice there is still some phosphorylation, so that kind of goes to demonstrate that mu-opioid receptors have a kind of cellular memory, if you like. So a naive receptor, which has never been exposed to opioids, will not respond in the same way as a receptor from a morphine-exposed mouse, for example. And that's due to these phosphorylation patterns, because some of the phosphorylation will remain even after recycling. And that's part of what we think, what scientists think leads to tolerance. And it's just important to note that obviously the phosphorylation patterns on this diagram are highly simplified. There would be a lot more phosphorylation at lots of different points, but we've just simplified it to a single P to represent this. So back to the aim of research approaches to maximize analgesia and reduce tolerance and withdrawal. There are a lot of upstream interventions which can provide more permanent solutions, which can maximize our own intrinsic pain relief mechanisms. So for example, you could use genetic engineering techniques such as CRISPR to target key mu-opioid receptor regulation genes, for example OPR-M1, which expresses the mu-opioid receptor. So in theory, you could edit the structure of the mu-opioid receptor to lead to a structure which would maintain your analgesic desirable properties, but not lead to dependence or tolerance. And there have been some studies which have found a few single nucleotide polymorphisms which could have the potential to do this. So there is definitely potential in the future to prevent or even treat addiction by permanently changing the structure of the mu-opioid receptor using genetic engineering. Some other techniques which maybe in the shorter term might maximize analgesia of endogenous opioids include using DENKIs. So DENKIs are these any molecules which modulate or inhibit enkephalinases, and enkephalinases are enzymes which break down enkephalins, which are an endogenous opioid agonist. So by blocking these enkephalinases, you have more intact enkephalin, which can bind to your mu-opioid receptor and produce analgesia, which is what we want. And very similarly, there's these receptors known as atypical chemokine receptor 3, which is it's atypical because it's a chemokine receptor, but it also binds promiscuously to a lot of different opioid agonists. So it kind of basically acts to sequester them and take them away from the mu-opioid receptor. Or it doesn't remove them directly, but it just means there's fewer agonists available. So if we were to design a molecule such as LIH383 to block this receptor, then that would leave more agonists available for the mu-opioid receptor. So that's the upstream interventions. If we look at the receptor now, there's a lot of research efforts going into trying to produce biased ligands. So this is based on this idea that if you can produce an agonist which selects for certain signaling pathways and not others, then you might be able to select for certain effects. So it's thought that G protein signaling is mostly responsible for analgesia, whereas arrestin recruitment is more responsible for withdrawal and tolerance and dependence and things like that. So the aim is to kind of design an agonist which favors this pathway. And so far a lot of those efforts have gone into trying to produce conformationally biased agonists, or using the chemical structure to favor certain conformations which favor the G protein signaling pathway. But in the future, maybe temporal or location parameters could be taken into account to target, say, specific neurons, or to target specific mu-opioid receptors on internal membranes to have a different effect. And we're essentially trying to mimic the patterns of endogenous mu-opioid receptor agonists, which don't cause tolerance and withdrawal. So yeah, the main idea is to just favor certain pathways over others. So downstream of the opioid receptor, a lot of efforts into reducing tolerance focus on phosphorylation patterns, as that's thought to be what causes tolerance, as I mentioned before. So one way of doing this would be to inhibit the G protein receptor kinases. So any efforts which go into developing these inhibitors could potentially be anti-tolerance drugs. You could also target arrestin recruitment by either sequestering arrestin or preventing it from being produced genetically, or blocking its binding somehow. So any interventions in which prevent arrestin recruitment would prevent internalization of the opioid receptor, so it could still be activated and prevent tolerance. Another intervention to prevent tolerance would be activators of protein kinase C. So this is a post-endocytic intervention because it occurs downstream of this endosome formation. And this is thought to reduce tolerance because in tolerant individuals, this pathway is down-regulated. So increasing the recycling of the mu-opioid receptor will reduce tolerance. So the difference between tolerance and dependence is that in tolerance you need more and more opioid agonist for the same effect, and in dependence you're basically addicted and you get withdrawal symptoms, and that's caused by the body trying to make adaptations to compensate for this chronic exposure. And your receptors are not meant to be activated all the time, so your body will compensate for this by upregulating certain pathways and things like that. So a few ways we can try to combat this would be to target the adenylyl cyclase pathway, which is upregulated in mice or humans with chronic opioid exposure. So drugs targeting this, or targeting some downstream effects of cyclic AMP, would be good targets to prevent dependence and withdrawal. Another downstream effect would be the mitogen activated protein kinase signaling cascade. and specifically the ERK cascade, which is upregulated in chronic opioid exposure and in withdrawal. So it's thought that if we could find inhibitors of this pathway, that could prevent withdrawal. So this is just a summary of all the mechanisms that we've talked about. A few things that we didn't mention though were targeting the G protein receptor kinase 5, which is thought to be important for reward signaling, and it's found to be upregulated in heroin addicts. So inhibitors of GRK5 could be used as anti-dependence drugs. Astrocytes are also important regulators of neuronal function and plasticity, and altered astrocytic signaling could be a major driver of dependence-like behaviors, so that could also provide another target for anti-dependence and anti-tolerance drugs. And finally, targeting the Glia inflammatory response. So there's a lot of evidence to suggest that the inflammation response in Glia could play an important role in dependence and withdrawal. So reducing that could provide good potential targets for drugs. And the ideal outcome, I guess, would be to have anti-dependence and anti-withdrawal drugs that you could take with normal opioid drugs like morphine.